All right, hi guys. So now we're going to move on to repressible operons, right? The repressible parts of the actual bacterial genome. Okay, so this is still a type of negative control. So if I just to remind you guys, essentially negative control, let me actually get to the spot where I'm at and change it. Negative control is basically the gene is expressed, right? And it only, like, the expression occurs all the time unless it is shut off by a regulator molecule. So for example, for the lac operon, it works unless it was shut off by the repressor, right? So that was a type of negative gene expression, negative control. But then when we looked at the CAP protein, at the catabolite repression, that was a type of positive control, because positive control is gene expression only if a regulator molecule directly stimulates transcription. So basically, I didn't realize I changed my camera, sorry. The the gene only gets transcribed when something is attached to it, when the cap, for example, was attached to it. So now we'll see that with both the negative and the positive control with repressible operons. So what we looked at was an inducible operon, that was a lac operon, where we had an inducer came in and that induced the actual genes to make the enzyme, okay, to be transcribed into the enzyme. A repressible operon is one that is usually on, and then the binding of a repressor to the operator shuts off the transcription. So again, a type of negative regulation. And the operon that we're going to be looking at is a trip operon. One way to really help distinguish which operon is inducible and which one is repressible is by seeing what the operon's structural genes actually are for. So if the structural genes encode enzymes that are necessary for digestion and catabolism, that is usually an inducible operon. If the structural genes encode enzymes that are necessary for anabolic pathways, so biosynthesis, so making things from scratch like amino acids or vitamins or whatever have you, those are usually repressible operons. And in fact, we're going to be looking at the tryptophan operon. This operon makes the amino acid tryptophan. So here we go. All right, so with the tryptophan, in order to make it, we have a particular enzyme called tryptophan synthetase. Now, it's a little bit weird because some textbooks say that there's one enzyme involved, other textbooks say that there's 3 enzymes involved. I don't really care that you remember how many enzymes are involved, but you do need to know that there's obviously, because it's an operon, because it's a cluster of genes, there's going to be obviously more than one gene that is important to allow for the tryptophan synthesis to occur in the first place. Those genes are TRPE, D, C, B, and A. So these genes right here encode for, in this picture here, they encode for 3 different enzymes. So they encode for 5 polypeptides, and then the polypeptides, some of them interact with others, some of them don't, and we form 3 enzymes. Your book states that these 5 contiguous genes are necessary to make 1 enzyme. Doesn't really matter, just know that there are 5 contiguous genes, right? And they're one next to each other, and all of the, since it's in an operon, all of them are under the same regulatory sequences. They're all regulated by the same promoter and the same operator. So if we see here, I believe you can see, even though it's not zoomed in, you have all of your structural genes, right? Just located side by side, right next to each other. And then right before your structural genes, so upstream of your structural genes, you have your promoter, your operator, and even something known as a leader sequence, which we mentioned and we talked about, I believe, in chapter 13, 14. I don't remember which chapter, but we'll go over what the leader sequence is in just a second. But that's essentially a part that is still transcribed, but it's not translated. So we'll get there. Okay. So what happens here is that since our tryptophan, this is basically our tryptophan is made. You can see in this image, you have your amino acid tryptophan is made. You have your raw precursors that is worked on by, an enzyme or multiple enzymes, whatever kind of complex that is encoded by these 5 contiguous genes. The precursor is made eventually into tryptophan. Now, when you think about it in the standpoint from a bacteria, the bacteria is not going to want to make its own tryptophan if it's already in an environment with tryptophan. It's just not going to want to do it, right? Because it's just a waste of materials, it's a waste of energy, it's using up all these crazy, we saw how complicated the processes of transcription and translation are. Why actually go through those processes if it could just simply gain the tryptophan from the outside? So in the presence of tryptophan, when the E. coli, when the bacteria has tryptophan around it, it's not going to synthesize the tryptophan. It won't. There's no need to synthesize it, and therefore it's not going to express these genes, these 5 structural genes at all. However, when it isn't in an environment with tryptophan, right? This does not find it in an environment. It is definitely something that it needs. Tryptophan is an essential amino acid. It's one of the essential amino acids. So it's going to activate its genes, express them, and make them into their corresponding enzymes to then make and synthesize tryptophan. Okay, so this is basically the concept behind it. It's going to shut off its genes when it doesn't need to make tryptophan because it can get the tryptophan from the environment, and it's going to turn on the genes when it needs tryptophan because tryptophan is absent from its environment. The tryptophan operon, the same guys who basically discovered the lac operon, so Jacob and Monod, those French guys that I can't pronounce the name of, right? They're the same ones who discovered the tryptophan operon. And it was actually a really good thing that they did because they realized that there was a lot of similarities between the trip operon and the lac operon, the one that we just went over. Except that even though it was very similar, right, there were some really key big differences. For example, the repressor in this case, you guys know that repressors bind to the operators, and when they do bind, they do, you know, whatever their name suggests they do, repress, they repress. So the repressor in this moment was, in this particular case, was inactive when it was alone, which is not the normal case in with the lac operon, and active when it was bound to something else. In this case, the thing that it's bound to is called the co-repressor, since the co-repressor helps the repressor actually repress, right? So the co-repressor is required for the repressor to bind to the operator and actually repress the operator and the transcription of that operon. So yeah, that's essentially how it works. I know right now it's just a bunch of words coming at you guys, but as soon as we look at it on paper, hopefully it will make a lot more sense. Okay, so I'm gonna change video right now. All right, so I have to stop the share. And here we go. Okay, so the tryptophan operon. I actually already forgot the order of the genes, but it doesn't really matter. I think it's tryp EDCBA, but correct me if I'm wrong, guys. Again, doesn't really matter what the structural gene orders are, just that we have 5 structural genes, which just underlines the fact that it's an operon, right? It's a cluster of genes. And so here we have, I'm actually going to draw in the leader sequence because it's very important when it comes to the operon. Okay, the leader sequence, then we have our promoter, and then upstream of the promoter, I'm going to put the operator. Okay, so the thing here, the thing with the tryptophan operator is that operon, it too has a repressor that binds to it and represses transcription and all of that. This is going to be found a ways away. Note how I do that break. That break just means it's a ways away from the operon here, the tryptophan operon. So it's somewhere else on the bacterial genome. We have this called TRPR. This gene is constitutive and it's always going to make our repressor protein. Our repressor protein is always essentially alive, or basically found as a protein. But whether this protein is active or inactive depends on what is bound to it. Okay, now with the trip operon, so remember this is basically our repressible operons. When there isn't any, when basically the co-repressor is alone, the repressor is alone. So there isn't any co-repressor. And in this case right here, the co-repressor is usually the end product of these genes. And we know that these genes together make tryptophan. That's the end product. So the co-repressor, I'm just going to write here, co-repressor is tryptophan itself. Okay, it's known as a negative feedback loop where the end product basically regulates and controls the beginning of the whole process. So the co-repressor is tryptophan. When we don't have the co-repressor, so C is missing, and the repressor is alone, in this case right here, unlike the lac operon, the repressor is going to be inactive. Okay, so if the repressor is inactive, it can't bind to the operator, so nothing's bound to the operator, and so transcription happens as per usual, like basically it's actually from the promoter, sorry. Transcription happens as per usual, and all of these genes are going to basically be made into the corresponding enzymes, and at the end, we're going to have tryptophan is made, which makes sense because if there is no tryptophan, right, our co-repressor is missing, there's no tryptophan, we want our enzymes to make tryptophan. So here, transcription occurs, and we make, or the bacteria makes, sorry, tryptophan. Okay, the other case scenario is we do have tryptophan. So this means this scenario is no tryptophan here, there is yes tryptophan. Okay, if there is tryptophan, that means we have our co-repressor. Tryptophan itself is going to bind to the receptor. The repressor. Okay, remember it's a co-repressor. So in this case, the tryptophan it co-represses the repressor's stuff. Whatever the repressor has to repress, that's confusing. It basically co-represses alongside with the repressor. It activates it. Okay, so when we have our co-repressor, when now we have something that is bound to the receptor, why do I keep saying receptor, guys? I'm sorry. The repressor is activated, and now because the repressor is activated, it binds to the operator and it stops transcription because it gets into the way. It bends all the DNA, etc., and it basically hides the promoter away from the RNA polymerase. So here the repressor is active and transcription doesn't work because since it's active, it binds to the operator and prevents RNA polymerase from working. So transcription stops, so we do not make tryptophan, but that doesn't matter because there's tryptophan already in the environment. And this is a tryptophan that went ahead and co-repressed along with the repressor. So it's pretty simple, really, but you just have to make sure you don't confuse this with the lac operon, because the lac operon is literally the opposite. The repressor is, when it's alone, is active, and the repressor, when it's bound to something, is inactive. That's the only difference, really. And then the rest basically works as itself. You just have to remember which form is active and which form is inactive in the repressor. So that's essentially how the tryptophan operon works. This is again a negative, it's a repressible system under negative control. And this way the bacteria doesn't make too much tryptophan when there is no need, and it makes tryptophan when there is a need, right? So again, look at these pictures because they're a lot better than mine. You can see again that you have these 5 structural genes. Oh, I got the order of the genes right. Anyways, you can see that these 5 genes are going to be made into one single polycistronic mRNA, and then afterwards that mRNA is going to be translated into the various components of this tryptophan synthetase complex. Right. So yeah, here it goes ahead and explains a little bit that the operator is called TRPO, the promoter is called TRPP, the repressor is encoded by a gene a ways away from the tryptophan operon. So far, they also made the little breakage sign. So far away from the actual operon itself, we have the repressor gene that is always constitutive and always active, right? And that's essentially it. So this is another type of basically initial transcription control, but with repressible operons that we have. The lac operon we also saw was transcription initiation control as well. Then we move on to, there's a different kind of control that works and basically manipulates RNA secondary structures. So we know that RNA is single-stranded, but we also know that the single-stranded RNA can basically form complementary regions within itself, right? It can base pair within itself and form little weird loops or stem-like structures that we also called hairpins, okay? So even these type of hairpins or secondary structures found in the RNA can give an added component of regulation to our genome, to our prokaryotic genome. And one of them is through a process known as attenuation. The other one is through a process known as riboswitches. So the use of riboswitches, which are specific elements found in the RNA leader sequence. Both of these are just ways to fine-tune the prokaryotic gene regulation, and they're used in addition to regulation of transcription initiation. So again, just underlining that regulation isn't usually as simple as just the repressor being active or inactive. There's a lot more stuff involved. We saw with the lac, it was more than just the repressor being active or inactive. We also saw that the lac necessitated positive transcription control through the use of the CAP, right? Through the use of the catabolite repression. And now we're also going to see that the tryptophan actually uses a process also in addition to the repressor active form or the inactive form. It also uses an additional process known as attenuation, okay? And to talk about attenuation, there's a couple of things to take into consideration. Yeah, a couple things to consider. Okay, so first off, I'm going to talk about the leader sequence just a little bit. So in my terrible drawing here, I'm going to stop the share so you can see this. In my terrible drawing here, you can see that this whole part right here, right after the leader, right after the promoter and before the actual structured genes themselves, we have a leader sequence. Okay, this leader sequence is right after the transcription point, the transcription start point. So we know that when RNA polymerase binds to the promoter itself isn't actually transcribed. It's just a place for regulation, as we're clearly seeing now, and it's a place for the RNA polymerase to actually bind to and start, unwinding the DNA to start transcribing it. But the transcription always starts at the +1 site. And even though the +1 site happens right here, we do have a stretch of the gene called the leader sequence that doesn't actually contribute to the polypeptide that will be translated. It's not actual coding gene. The genes are these ones right here, E, D, C, B, and A. This is going to be the coding part. So when we make our mRNA is going to have a little bit of that leader sequence. So we're going to transcribe a little bit of that leader sequence, and then we're going to have the main coding part, the actual structural gene part, the cistron part right here. Okay, so that when we translate this mRNA, only this purple part is going to be translated, essentially. This leader is more here. This leader sequence doesn't really get translated and made into any substantial protein, okay? It's mainly there just for regulatory reasons. And it's extremely important, because in this leader sequence, we have specific attenuation sites, and we also have a small little peptide known as the leader peptide or the trip peptide. It depends on, again, the textbook. There's all sorts of words that you can use, but it doesn't really matter. I'm going to call it the leader peptide. So we have a initial part right here that encodes the leader peptide. And strangely enough, this leader peptide, when translated, so it's a very minuscule protein, when translated, not exactly the first, but the beginning, at least at the very beginning, it has a couple of amino acids, and it's two really close. These two amino acids, the two first amino acids, are tryptophans themselves. Okay, so you need basically tryptophan in order to translate this leader peptide sequence into the leader peptide. Okay, so you need two tryptophans. I'm going to color them black. Okay, two little tryptophan residues. Downstream of the leader sequence, we have a few regions. We have region 1, region 2, 3, and 4. Okay, these are known as attenuation sequences. These are attenuation sequences. And the thing about these particular sequences is that as soon as they're transcribed, they're going to form particular kind of stem loops. They're complementary to each other. They're going to form particular stem loops. What are those possible stem loops? So number 1 and 2 can come together, right? And then if regions 1 and 2 come together when they're transcribed, so when transcription happens, I know here it's a very nice linear mRNA. That doesn't necessarily happen if we have complementarity, right? So 1 and 2 can bind to each other, and then 3 and 4 will bind to each other as well. So that's 3 and 4. And what happens here is we call this loop the pause loop, and this loop right here we call the terminator loop. Dun, dun, dun. Okay, the terminator loop, which if formed will terminate transcription. The other possibility is that region 1 will be temporarily, once transcribed, will be transcribed first. Then region 2 will be transcribed. But as region 2 is transcribed, usually 1 and 2 pair up. But say there's something that's covering region 1, region 2 is going to be transcribed, then followed by region 3. And so region 2 and 3 are going to pair up because there is no 1 for region 2 to bind with. So region 2 and 3 are going to pair up. and then region 4 will be made. So we have region 1, 2, and 3, and 4. If this particular stem loop gets made, this is known as the anti-terminator loop. And as you can imagine, this does not it basically is the anti-terminator. So it's the opposite of the terminator loop. It does not terminate transcription. Okay, so I know this still doesn't make any sense right now, but it will in just a second. So attenuation basically is something that only happens in bacteria, because bacteria have this have co-translation and co-transcription. Essentially, because they don't have a nucleus, their DNA is found free floating within the cytoplasm. When the DNA gets transcribed into mRNA, as soon as that mRNA is made, a ribosome can latch onto the mRNA and translate it immediately. So translation and transcription are basically side by side, okay? This is really important because if translation and transcription are side by side, so like the ribosome for translation is side by side, a couple of things, some regions of the mRNA may be hidden at certain times and some may not. So let's look at the possible scenarios that we have. Say we have a scenario where we have a lot of, so yes to tryptophan, an increased amount of tryptophan, okay? This is our possible scenario. Remember here, protein synthesis and transcription are coupled. Okay, so the ribosome is going to be trailing behind the mRNA or the RNA polymerase very, very closely. Okay, you can actually even see it in the picture here, I believe. Where is it? You can see that this is a ribosome. Ribosomes are necessary for translation. So it's found right after the RNA polymerase. Still not that great of a picture. Hold on. So this is a better picture that I found in one of my other slides from another course. So we have the RNA polymerase, and as you can see, right behind it, trailing basically behind it is the ribosome, okay? And so this is extremely important to remember when it comes to tryptophan operon attenuation, because if this wasn't the case, we just have the RNA polymerase by itself. In fact, let's actually see that, that first scenario. So if we had that scenario when we have protein synthesis, right? So our ribosomes are working, but the protein synthesis is slowed down, which actually happens quite normally in bacteria, depending on whether they have to like survive the environment or not. So when protein synthesis has slowed, the ribosome is nowhere near the RNA polymerase. So transcription and translation are not tightly coupled, okay? The ribosome's far away. It's really hard to keep up with the RNA polymerase because protein synthesis has slowed down. So when that's the case, when the RNA polymerase reads the DNA, and as it reads the DNA, it's transcribing the RNA, and it goes into that trip-L sequence, right? That trip-L, the trip-L, remember, is that leader peptide. It makes that leader peptide and makes into mRNA, and then it goes through those attenuation sequences. As soon as it makes like attenuation sequence region 1, and then it makes region 2, so it's about to make 3 and 4. Region 1 and 2 have already made, so they bind up, have already been made, so they bind together, they form a loop. And then the RNA polymerase makes region 4, and then the 4 doesn't come before 3. The RNA polymerase makes region 3, and then it makes region 4, and then it keeps going. Actually, it keeps going, tries to keep going. But as it makes region 3 and 4, those pair up, and you guys know that the region 3 and 4 are known as a terminator loop. This terminator loop terminates transcription. It basically destabilizes this entire transcription initiation complex. It kind of bends everything really weirdly. It makes the RNA polymerase kind of very loosely pull away from the DNA, and then eventually it actually ejects the RNA polymerase right out. So when protein synthesis has slowed, right? Because there's just something's going on. Protein synthesis is slowing down. Transcription of the tryptophan operon doesn't work, which makes sense because why make your amino acids, the building blocks of protein, if the synthesis of protein has slowed in the first place? So that makes sense. The next scenario, the next two possible cases, depends on, we're considering the possibility that protein synthesis is working as normal. So transcription and translation are coupled. So our ribosome is closely next to the heels of RNA polymerase. Is that how you say it? It's on the heels of RNA polymerase. So the ribosome is on the RNA polymerase's heels. Yeah, I got that right. Okay, so they're really close to each other. Okay, and this dictates how how and which regions actually are able to be formed, which regions are going to be complementary, they're already complementary to each other, but which regions can form which stem loops. So in the case that tryptophan levels are low, okay, tryptophan levels are low, the ribosome pauses at the tryptophan codon, right? Do you remember how in my scenario right here, we had a leader peptide, okay? We had a leader peptide, hold on. A leader peptide that when it was translated, it needed 2 tryptophan. But in this case here, there is low tryptophan level. So this means that our tRNAs, because remember this is translation, our tRNAs that are normally charged with tryptophan don't have tryptophan, they're uncharged. And so the ribosome basically stalls right here. It waits until those tryptophan levels, those tryptophan tRNAs are coming into play. Does that make sense? So until like the tryptophan tRNAs come in and actually allow for the continual translation of the leader peptide. So our ribosome stalls a little bit. Our ribosome stalls because it's just waiting for those tRNAs to have those charged tryptophan, which are very scarce because our tryptophan level generally in the cell is quite low. So as our ribosome stalls, the rest of the RNA polymerase keeps going, right? Because remember, RNA polymerase is simply transcribing. RNA polymerase is simply transcribing. So, yeah. So when the ribosome stalls, this blocks region 1. So if you see here, the ribosome is stalling. It's waiting for this tRNA with the tryptophan to come in. And as it's waiting, it's situated right on top of region 1. And then RNA polymerase is going to continue and make all the other regions. So it makes region 2, it makes region 3, and as it's making region 4, 2 and 3 pair up. And we know that when 2 and 3 pair up, it forms the anti-terminator loop. And this anti-terminator loop is able to inhibit the termination of transcription. It's a very stable kind of conformation of the mRNA, and it allows the RNA polymerase to go ahead and transcribe, which is perfect because we want transcription in this case to happen. We want transcription to happen so that our genes that encode the proteins, the enzymes that make tryptophan are made, which is perfect again because we have low tryptophan in the first place. So this works out. However, when we have a high amount of tryptophan, the ribosome does not stall at region 1. It doesn't stall at region 1 because that leader peptide that needs those tryptophan residues is going to get them really quickly, right? The ribosome goes over region 1. It's going to need those two tryptophan residues that it needs to make the leader sequence, right? And it's just going to go right over it. So it's going to translate without stalling. And as it translates without stalling, it moves on to region 2. And so as it's in front of region 2, this RNA polymerase makes region 3 and region 4. Region 3 is not going to pair up with region 2 because the ribosome is over region 2. So it's going to make a region 3 and 4 complementary stem loop. That stem loop is the terminator loop. And it's going to destabilize everything. It's going to destabilize that initiation transcription complex, and RNA polymerase falls off. This again makes sense because we want RNA polymerase to fall off because we don't want it to go ahead and transcribe and make those enzymes that make tryptophan when we already have a high amount of tryptophan in the first place. Attenuation is a tough one. It's definitely very tough to remember and to think about because there's a lot of different sequences and regions and peptides and stuff to follow. And in addition, you're following both transcription and translation happening at the same time. I don't think your book does a very good job at explaining it, but it may do a good job for you. Who knows? It's different for everyone. But just know that, ask me questions if it's something that you don't understand, because it is a very difficult concept to remember. I also think I shared the wrong, yep, I did. I shared the wrong PowerPoint. Okay. No, I didn't. I shared the correct one. Sorry, guys. So this is attenuation, and this is just an extra. So our tryptophan operon can be worked on by the repressor action, right? That negative control. And now we can see we also have this added regulatory control of attenuation as well when it comes to not simply transcription initiation, but actually a little bit past transcription initiation, a little bit into transcription elongation right now. And you can see how it's basically dictated by the secondary mRNA structures that are made, right? You can see like the stem loop, these different stem loops are made, and whether we have a terminator loop or an anti-terminator loop, that allows for either the continuation of transcription with an anti-terminator loop or the inhibition of transcription with a terminator loop. So just to show you that the secondary structures that were found in mRNA can indeed control how transcription proceeds. Another thing that we have, another type of regulation that we have that still involves secondary structures in mRNA are known as riboswitches. These are just regulatory sequences found within essentially the 5' untranslated region of the mRNA, which is pretty much the leader sequence again, right? So right in front of the genes, the cistrons that are found on the mRNA, we have a place that does not get translated. It's the untranslated region, so 5' untranslated region. There's always, in mRNA, there's always a 5' untranslated region, the genes themselves that are going to be translated, and then a 3' untranslated region at the end. But in the 5' untranslated region, which is essentially the leader sequence, we have sequences of these mRNA elements called riboswitches, which can be turned on or off. And depending on whether they're on or off, they assume different kinds of secondary mRNA structures. So they make different kinds of loops, right? So these guys are capable of binding with small molecular ligands. These ligands tend not to be proteins, so they're more like effectors. They usually are things like nucleotides or amino acids. So something like flavin mononucleotide (FMN), that's definitely a ligand that can bind to these riboswitches. And their activity, their activity, or basically the transcription activity, can then be controlled by this ligand binding to the riboswitch or not, right? So the binding of the ligand to the riboswitch causes a conformational change in one domain of the element, and this then induces another change at a second RNA domain, which then forms a transcription terminator structure. So when the ligand binds, here we go. When the ligand binds, it forces the different riboswitches in the gene, in the 5' untranslated region right before the gene in the mRNA to adopt specific kinds of stem loops. And one of these stem loops is going to be the terminator stem loop. And so the ligand binds and it's going to terminate transcription because it's forced these riboswitches to form a terminator stem loop. If the ligand is not present, these riboswitches are normally going, these elements in the 5' untranslated region are just normally going to form an anti-terminator loop, just like so. And that's fine. But in the presence of a ligand, the ligand forces different secondary structures to come about. And one of the secondary structures is a terminator loop, which terminates the transcription. Anti-terminator loops do not terminate transcription, and therefore they allow for the transcription of the downstream genes to be made, to be continued to be done. That's essentially how riboswitches work. They literally are like a switch, the ligand binds or not. The specific sites that the ligand binds to is called the aptamer, and the expression platform is just basically the platform where we have the riboswitches, where all these possible secondary structures can be made. So either the terminator loop or the anti-terminator loop, one of those structures. It's pretty simple, really. There's nothing too complicated about them. Here it tells you that some ligands of riboswitches include amino acids, purines, like those nucleotides, like the flavin mononucleotide I told you about, vitamins, metal ions, etc. There's all sorts of different things that can bind to it. This has been prevalent in bacteria, but it can also be found in archaea, fungi, and plants, and even in some animals too, apparently. So that is it for prokaryotic regulation. It's quite complicated, right? We looked at initial transcription regulation, and we also looked at a little bit of post-transcription regulation, or when transcription has already started, right? And we're already in that 5' UTR mRNA, and there's secondary structures that can form directly into the mRNA that's just been made. And that includes things like attenuation and riboswitches. Okay. Actually, hold on. I'm going to make you guys a little concept map to kind of go over all of this information, because I don't want you to be confused, even though I just hope you're not confused. So what we saw, we looked at 2 different kinds of operons. We looked at the inducible operons and the repressible operons, okay? And then the inducible ones. And we saw that both of these can undergo different kinds of, like, different repressors can bind to them, okay? Of course, though, the repressor for the repressible operon is active with a co-repressor, and the repressor for the inducible operon is only active without the inducer. So we need to remember the LAC is a type of inducible and the tryptophan is a type of repressible. So you have repressors that act, and that's our negative form of transcription regulation. In addition, we saw that with inducible operons, with the LAC operon, we also have catabolite repression, that CAP. That's a type of positive transcriptional control. And then we saw that with the tryptophan, we can also have attenuation, right? But attenuation, something that's different about attenuation compared to repressors and the CAP is that attenuation happens post-transcription, meaning not post-transcription, like not towards the end of transcription, but post the initiation of transcription. Well, these guys right here basically work and regulate right at the beginning of transcription, and they either allow transcription to happen or they stop transcription from happening altogether. Okay? And then the other thing that we noticed, that we talked about, is that if we look at, again, post-initiation transcription, after the initiation of transcription, we have these riboswitches, which are found, if I draw out the mRNA, right, if this is my mRNA, my AUG, my start codon for the genes is going to be somewhere here, and then before it is the 5' untranslated region. In here, we have these elements known as riboswitches, and these riboswitches can form all sorts of weird stem loops. And depending on the stem loops that they form, they can either form one stem loop that is the anti-terminator, or 2 stem loops, one that is bound to the ligand, right? And that ligand binding is going to form the terminator loop. This terminator loop terminates the rest of transcription. So none of this is going to be made afterwards. So these riboswitches occur in all over different kinds of bacteria. They're especially, when it's in terms of transcription level, they're especially prevalent in gram-positive bacteria. So riboswitches can happen with everything. And that is basically it. So I'll see you guys in the next video.